sorry to the 3 of you who came at 7 for the online portion. Canvas conferences wasn't working for me, so I'm going to talk to IT and get that figured out for next week. And for this week, rather than missing another week of review with you, I'll just record this video, and it won't be as interactive as I had planned, but the beauty of videos like this is you can pause it as we go and kind of interact on your own, if that makes sense. Feel free to pause it, see if you know some of the answers to the questions, and really get involved, because the more you get involved with these reviews and with studying, the better time you're going to have remembering things. And so I encourage you to really pause every so often. Make sure you know what is going on, because these are topics that students generally have trouble with, and it's important to know for the test and just for normal life. So we'll go ahead and get started, and I thought we'd start with the scientific method, because the scientific method is huge. It's what nearly all research is based off of. So we'll go through it, and then we'll have a perfect spot to kind of pause, and maybe you can create your own experiments or an experimental design, meaning maybe take a couple minutes and write out a possible experiment. Make sure you understand the steps. So we'll get going in here. So the scientific method has really 5 main steps. First, we have to come up with a question, and then a hypothesis. Then we'll experiment and analyze the data and form a conclusion. So starting with the question. A question is most effective in science when it's specific. For example, what protein does the antibiotic name an antibiotic target? And so it's specific, it can be measured and tested, and then, like Dr. Rosen was talking about in class, there's a difference between scientific-based science and hypothesis-based science. Scientific-based science is more descriptive. It's observations. For example, migration patterns in birds. There's also hypothesis-based science, which seeks to explain those things. For example, why do birds travel south for the winter? And that leads us to create, wrong direction, sorry, to create a hypothesis. And there's a difference between a good hypothesis and a mediocre hypothesis. A good hypothesis, again, needs to be specific. It needs to be measurable. It needs to be something that you could easily collect data to either accept or reject your hypothesis. A lot of times it's formed in an if-then statement. For example, if I were to dye my hair red, then I would get more people staring and looking at me as I walk by. Now that seems like a silly example, a silly hypothesis, but it's measurable. It has defined parameters, which are dyeing my hair red, and it has something easy to calculate, the number of people that look. A good hypothesis is also repeatable. Could I do this experiment again and again and get the same results? Which moves us on to the actual experimentation. This is where the research is performed, where data is collected. And we move on to setting control groups, if necessary, or experimental groups. A control group is very important in determining whether or not what you're seeing is an effect of the environment or the variable you're studying. So in the trans fat example, there was a group. The experimental group was those who had had a heart attack in the past. The control group were of similar ages, similar backgrounds, but the only difference was that they didn't have a heart attack. The best control group is one who only has one variable, one thing different than the group that you're studying. In this case, the only thing different was that they had not had a heart attack in the past. And that data that you collect will help you to form your conclusion. Is your hypothesis proven? That's a trick question, because hypotheses are never proven. They are accepted or rejected. In science, we don't like to use the word prove when talking about the scientific method, because it takes a lot of evidence in order to prove something. That goes more along lines with scientific theory or scientific law that Professor Rosen was talking about in Lecture 1. Rather, when we're experimenting and testing hypotheses, our evidence either supports our hypothesis and more testing would be done, or it rejects or it nullifies our hypothesis. And we go back to the drawing board and maybe figure out another question, another possible reason behind what we were studying. And we want to make sure that our conclusions and the conclusions that others draw are reliable, especially if we're going to use them as a basis for our learning and possibly papers you write down the road. You want to make sure that they're peer-reviewed and that they've been used often, meaning others have looked at the research, seen the methodology, and accepted it as scientific. So, with those things in mind, create your own experiment. Write up a makeshift experiment, and if you want to, email it to me, and I'll look and see that either you're understanding it well, or maybe we need to clarify some more with you. So that's the scientific method. We'll now move on to our next subject, which is the basic chemistry needed to understand biology. And again, we've got another. So our next portion is the basic chemistry, shown here by this presentation. Alright. So this is basic chemistry, and what we're looking to do is break down life as we know it into its very smallest pieces. And with this, we're going to do a lot of comparing and contrasting. We're going to figure out how the basic units of chemistry are similar and different. And in order to do so, I thought we'd just focus on one molecule, one compound, being water, or H2O. So, is water a molecule or a compound? That's also a trick question. Water is both a molecule and a compound. And that's because water has two different elements in it. And a compound is a grouping or cluster of one or more atoms, well, of two or more atoms, that have at least two different elements in it. Now, a molecule can be two atoms of the same element. Maybe two oxygen atoms would form a molecule. Whereas a compound, like I said, have to be at least two different. So, looking at the example of water, we have two hydrogens and one oxygen. With there being two hydrogens and one oxygen, that makes three atoms, which makes it a molecule, right? It has at least two atoms in it. It's also a compound because it has both oxygen and hydrogen, two different elements. And for all of these compare and contrasts, I'll make a worksheet for you to kind of go through and better analyze the similarities and the differences of some of these. So our molecules and our compounds are the biggest unit of chemistry that we're going to talk about tonight. And those are broken down even more into hydrogen and oxygen. Now, hydrogen and oxygen are both elements. Elements meaning that they are substances that can't be broken down into any smaller subunits. They are only hydrogen and only oxygen. Now those elements are made up of atoms. Atoms are the single smallest unit of chemistry, at least of the chemistry that we'll be talking about. So hydrogen is 1 proton and 1 electron. That's it. Nothing else. That makes up a hydrogen element, or a hydrogen atom. Oxygen, on the other hand, has its own protons and electrons in its given ratio, and they make up its elemental makeup. And again, with these on the compare and contrast, that will be included in the worksheet that I'll create for chemistry. So now on to the parts of atoms. What makes up an atom? Now an atom, like we were talking about a little bit on the last slide, is made up of protons, neutrons, and electrons. A proton has a positive charge. Both start with the letter P. That's how I remember it. And the number of protons determines the number of the atom, or the atomic number. Hydrogen being 1, etc. So then, which of the parts of an atom have equal numbers? Well, like Dr. Rosen pointed out, protons and electrons have the same number in any given element. So if the atomic number is 1, then it'll have 1 proton and 1 electron. This, of course, is only for molecules, or atoms, that are non-ionic, meaning they have a balanced charge. There's no net charge on them. So protons equal electrons. They don't have the same charge, but they have an equal and opposite charge, meaning 1 positive for every 1 negative. And inside the nucleus of that atom are the protons and the neutrons. That's what makes up the weight of an atom. Electrons don't have that weight. So again, equal numbers, protons and electrons. That's going to be something really important for the test. And I'll put the rest of this on a compare and contrast worksheet again. And I really encourage you to go through and fill in these worksheets. Really use them to study. There's so much information here, and you'll want to help yourself study. You'll want to classify and bunch information in any way you can. So those are the parts of an atom. And again, that's the smallest unit. And we'll tie it back into this iceberg analogy, where everything underneath the surface, or everything we can't see, meaning the parts of atoms and elements and molecules, those are all so much larger in number than what we see by our naked eye. So we're going to go back through and review. We've got atoms and elements excuse me, that are larger, and then molecules and compounds, which are even larger than that. And they all make up the basic chemistry. And we see this iceberg, we see that the visible portion, like I was saying, that water that we see, is quite small in comparison to the vast amount of components that make it up. And that's the beauty of chemistry, is that it opens our eyes to so much more that's out there than we even know. A great deal. And with that being said, I'll have the worksheets up and we'll move on to our last little portion. I'm not sure how well it'll work on here, so I may just make it into a worksheet for you, but we've got here a picture of a eukaryotic animal cell. Eukaryotic meaning, of course, that it's a larger cell, it's more complex, it has organelles or membrane covered pieces inside of it, and a true nucleus, or a nucleus with a membrane around it. And you'll see here that I've labeled all of these different pieces, but some of them are wrong. Some of these labels are incorrect, and I did that on purpose. What I wanted each of you to do tonight is to correct the errors, meaning test yourself. See if you can identify which of these are in the right place and which are not. See if you can tell by sight which component of the cell each of these is, and also you'll want to know what the purpose of each of these organelles is. Dr. Rosen talked about them a little bit in lecture, and again I'll make some sort of a study sheet for you so you can correct these errors, but also so you can learn the functions of each of these. I'll also put a plant cell and a prokaryotic cell that you can look at and determine where the errors are, and really give yourself practice, because the more practice you do now, the easier it will become test time to recall this information. And again, sorry for those of you who came to lecture, or not lecture, but to the review session and weren't able to do the conversation like we had planned, but I'll talk to the IT people and figure out what's going on, and hopefully we'll be able to get that worked out so that we can have a normal review session on Canvas Conference next week, next Thursday, again at 7, and we'll be going over the organic chemistry portion, and as well all the material that's big and important from week 3's lecture. Have a great week, and I will see you next Thursday.